Hello guys, and welcome back to Excel Academy. So today what we're going to talk about is different modes of replication. So till now what we have discussed is like the linear double-stranded DNA replication, and we have also talked about how in the case of prokaryotes it is happening, right? But this is something which is the rolling circle model of DNA replication. Even this is happening in few cases. So just let's talk about this rolling circle replication. Have you guys heard like about? Do you guys know what is conjugation? कभी सुना है what is conjugation? So understand that this is going to help in conjugation. So what we are going to study now, it is actually a part of conjugation only. Okay. So let's see how it is becoming a part of this. It's very simple. जब इतना बड़ा DNA replication topic हमने समझ लिया है, तो this is nothing in front of that. Okay. Okay. So replication in eukaryotes is bidirectional, and this is and जो हम अभी discuss करने वाले हैं, that is the rolling circle. Okay, this is unidirectional. So what does it mean? Only in one direction it is going to be there. So इसमें as such replication fork नहीं बनता है. Okay, so there is no replication fork formed. Okay, so or this happens only in the case of the circular plasmid of bacteria. Circular genomes only. Linear DNA में ये नहीं हो सकता. नाम से ही पता चल रहा है. It's a rolling circle. तो roll होता हुआ. So that is only possible in the case of a circular DNA, right? Now how it is going to initiate? It is going to initiate by the action of helicase, topoisomerase, and SSB. Now let's see. This is a bacterial plasmid. Okay. Now, bacterial plasmid, you can see that you have got the helicase over there, topoisomerase is there, and single-stranded binding proteins are also there. Once again, yeah. So how this is being started? There are certain proteins like Rep A, आपने सुना होगा. We have studied that replication A protein is there, which is actually starting over here. So what do we see is that they make a nick. Okay. So what will happen? You will get a 5' phosphate and a 3' OH end. Is that so simple? Hai, clear hai, right? The understanding is clear to this. So, and this is happening only in one of the strands. Only one strand is being nicked over here. Okay, nicked hua hai. Now, you have got now OH and you have here 5' phosphate. So we know that to the free OH only, the incoming nucleotide can add, right? Remember our SN2 mechanism, nucleophilic attack, vagera, vagera. Okay. Now, but which one is going to act as a template? कुछ idea है? कैसे होगा? Look it over here. So what will happen then? Over here, you have got the OH group of the broken strand, right? Now, यहाँ पे ये जो strand है, okay? आपका जो 5' end जो है, that will be getting unwound, वो separate होता जाएगा, okay? And DNA polymerase 3 is going to add new nucleotides at the 3' end. तो क्या हो रहा है? जैसे जैसे आपका 5' end वाला ये जो linear DNA है, it is getting removed. So this is going to act as a template now. अब हमारे पास template भी है, 3' OH भी है, DNA polymerase also we have. So there is no requirement of primer, right? Because to this 3' to this 3' OH only will act as a primer, and to this your DNA polymerase is keep on adding the new nucleotides. Strand कौन act कर रहा है? आपका नीचे वाला, right? Where this 5' has been removed. Is this much clear to you guys?
Yes, ma'am.
Okay. तन्वी समझ में आ रहा है क्या हो रहा है यहाँ पे?
Yes, ma'am.
Okay. Now let's see. See. Okay. So what will happen towards the end? Towards the end, what will happen then? So completely your 5' वाला जो था linear, it will come out, right? And over here, this is going to have its complete replication 3' OH. Last में क्या होगा? Ligase आएगा and reseal कर देगा. And there is no question of primer removal, right? हमें उसकी कोई टेंशन ही नहीं है यहाँ पे. सिर्फ लास्ट में एक गैप रहेगा जो आपका ligase आके सील कर देगा. ठीक है? Now, what about this? This linear DNA is now is going to get circularized. Okay, and अभी इसमें आपका replication शुरू होगा. Clear? So overall अभी क्या हुआ? पहले आपके पास ऐसे था, okay, and the outer one was removed like a linear. This got circularized. Now to this you have started the another round of replication. So what is happening? At the end I am having 2 plasmid, correct? Clear hai?
Yes, ma'am.
Okay, this is another figure. Okay, same cheese hai but another figure. So you can see it over here. A nick is made. So that nick is made with the help of the rep A. This is double stranded origin. यहाँ पे आपका single strand origin है, ठीक है? Then what is happening? SSM loop structure is formed. Then it is going to make a nick over there. Then finally आपका जो 3-OH है वो expose हो जाएगा. आपका DNA polymerase आएगा and will start doing the replication. So when this is done, the outer strand is getting released as a linear. फिर वो circularized हो जाएगा. Then after getting the circularized, another one will come and it will start doing the replication. ठीक है? Only one thing what you need to remember over here is that जो DNA आपका linear हुआ था, there you are going to require a RNA primer. है ना? Because there was nothing to start. So you need a RNA primer over there, and then the polymerase will come and do the synthesis. Okay. So how many times the RNA primer is required in a rolling circle replication? अगर मैं आपसे पूछूं, so what will be the answer? Yes. How many times? In one complete rolling circle replication? Yes, it's one times only. Okay. अगर आपके दिमाग में क्वेश्चन ये आता है कि origin कहाँ से शुरू है, so don't worry, you have got the specific sequence for that. Okay, so before starting you have got the double strand origin, and the next round आपका आता है like single, this is present, so you also have a single strand origin. So वहां पे आके आपका polymerase will start, the RNA primer will come and it will start the replication. Is this much clear? Okay. So don't think guys यहाँ पे बाकी चीजें required नहीं है, यहाँ पे भी हमें बाकी proteins लगते हैं. It's just that it's a simple version of DNA replication. Okay, and this is happening. Now what we have just learned now is happening in the case of the conjugation. So conjugation हम कब कौन से bacteria में सुनते हैं? Like in which case we have heard about the conjugation? Have you heard of F plus F minus bacteria?
Yes.
Yes. So there. So you can see that there is a F plus cell, donor bacterium is there. So they have got the F factor. So they form a, so during conjugation, what happens there, cell wall gets merged, and then you know that over there, again, one strand is going over there. That is your 5' जो हमारा जो linear DNA जो बन जा रहा है, ठीक है, upper outer strand. So this will complete its replication over there. This will go to the second one, F minus में जाएगा. वहां पे this linear DNA, the single stranded linear DNA is getting circularized and it will start the second round of replication. So at the end, what is happening? The donor recipient bacterium is also becoming a F plus. Okay. So this is happening only in such cases of rolling circle replication. Otherwise, the normal chromosomal DNA जो है, host का जो chromosomal DNA है, it is going to have the normal आपका replication. Okay. This is only in the case of plasmid DNA or the F plus, F minus case में. Is this much clear? The rolling circle DNA?
Yes.
Okay. So I just hope you guys will get the answer also. अगर इस पे base कोई question आया. So the proteins which you need to remember is Rep A. Okay. Rep A, which goes in binds to the double stranded origin, makes a cut over there, and then you have got the 3' and the 5'. 3-OH में you start adding the new nucleotides with the help of the DNA polymerase 3. Okay. 5' जो है वो आपका बाद में linear single stranded linear DNA बन जाता है, which later gets circularized. Okay. Now you can see that anyways, the helicase, the other proteins are also required. Okay. So don't think that over here the helicases or the topoisomerases are not required. It is required. Now, the next case, what will happen? Then you have got the single-stranded DNA to which the RNA primer will come and the DNA polymerase will start making the new strand. That is the second round of replication is happening. Okay. Then finally, at the end, what you're getting is closely, covalently closed circular DNA is formed in both the cases. Okay. So I'm sure this is done, and I guess I have got a video also for this. Let me just share the volume of this 1 second. Yeah.
In this image, the DNA from a bacterium has been gently released from the cell. Most of the DNA is from the bacterial chromosome, but small circular DNA molecules called plasmids also exist. For many plasmids, replication is not tied to chromosomal replication. Many plasmids replicate autonomously by a method called rolling circle replication. In rolling circle replication, a replication initiator protein called Rep-A binds to a section of the double-stranded DNA called the origin of replication, or ORI. Rep-A is encoded by a plasmid gene. Rep-A nicks one strand of the DNA and holds on to the 5' end of the strand. The 3' end, with its free hydroxyl group, serves as a primer for a host DNA polymerase to begin to replicate the intact complementary strand. The Rep-A initiator protein recruits a helicase that unwinds the DNA. As the DNA unwinds, it becomes coated by single-strand DNA-binding proteins. As replication proceeds, the nicked strand, which continues to be covered with single-strand DNA-binding proteins, progressively peels off until replication of the intact strand is complete. The two ends of the nicked single strand are rejoined by the Rep-A protein and released. DNA ligase seals the nick in the double-stranded molecule. The single-stranded DNA can now be replicated. A region of the DNA becomes looped, allowing RNA polymerase access to the DNA to form a primer. Host DNA polymerases use the primer as a starting point for the synthesis of DNA. Finally, DNA ligase seals the remaining nick, resulting in a double-stranded plasmid. Each of these plasmids can undergo replication again by the same method.
So clear? Any doubts? Shruti, you have joined late, but I hope you got the idea. We are discussing about the rolling circle replication. Yes? Yeah.
Yes.
So is it clear, Akshaya and Tanvi? It's very simple concept, right?
Yes, ma'am.
Okay.
Yes, ma'am.
So, yeah. So now let's talk about the theta replication. The theta replication, kuch nahi hai. It is just the same which we have studied in the case of bacteria. Okay. So you can see that both the sides of origin. See, you have got the origin, replication fork is moving and it is moving in both the direction, right? It's just that in the electron micrograph, it was looking like a theta. Okay, and that's why it was known as the theta replication. At the end, you got the two chromosomes which are like catenated, and with the help of the topoisomerase 2, it will get decatenated. Simple. वही अपना bacteria वाला ही है. Okay. Now, the another important topic is how the mitochondrial DNA is getting replicated. So we have talked about prokaryotic. अभी हमने plasmid भी देख लिया. We also have talked about the eukaryotic DNA replication. Now, what is left is mitochondrial DNA. And this is also true for chloroplast DNA also. Okay. So we know that in the case of mitochondrial DNA and the nuclear DNA, okay, we have got the, what I mean to say is that there are certain proteins which are required by the mitochondria, but are synthesized by the nuclear gene. Okay. So we know that in the case of the ETC, that is electron transport chain, there are so many complexes are there. For example, complex 1 is there, 2 is there, 3, 4, and then at the end we have got the ATP synthase, right? Which is sometimes also known as your complex 5, right? So out of these, there are many complexes. For example, complex 2, which is synthesized by the nuclear genes. Okay, so when it is synthesized with the nuclear genes, what does it mean? It means it is getting the translated in the cytoplasm, and from the cytoplasm, it is getting translocated to the mitochondrial matrix. If you remember, the complex 2 is also known as your succinate dehydrogenase complex. And where else you have heard about the succinate dehydrogenase? और कहाँ सुना है हमने succinate dehydrogenase? Yes. Which cycle it is present? I gave all the hints. Yes, it is in the Krebs cycle. Okay. So we know now that this succinate dehydrogenase is present, and the Krebs cycle takes place where? In the matrix, correct? But your ATC takes place where?
Mitochondria.
Arey Baba, this is also a part of mitochondria.
Membrane.
It is taking place in the intermembrane space. Sorry, in the membrane space में वहाँ present है ATC, right? It is present on the inner membrane space में आपके वहाँ पे. Okay, so you have got the mitochondria outer, inner आपका ये ऐसे crystal रहता है. On this you have got your ATC complexes. Okay, now this is your matrix. So यहाँ पे आपका succinate dehydrogenase है, but whereas your complex 1, 3, and 4, 5 are present in the, on the inner membrane space. Okay. Okay, so you can see that it is so difficult for a cell. जबकि काम mitochondria का है, but who is starting that work? Nuclear genes are starting. So from the nucleus, it is going to get transcribed, then translate होने के लिए वो cytoplasm में आएगा. Cytoplasm से आने के बाद it is getting translocated to the matrix of the mitochondria, right? And it has to cross the outer membrane, inner membrane, and then it is going into the matrix. So there is a very good protein trafficking or protein sorting, which we'll be taking during our cell biology. So there I'll show like how these proteins are getting translocated to the specific organelles. Okay. So abhi thoda sa idea mila. So what I want to say is that even though we are going to talk about the mitochondrial DNA replication, many genes are getting transcribed by the nucleus. Okay, and then they are translocated to the mitochondrial organelle so that the mitochondria can function. Okay. Okay, so look at over here in this figure. So the main part which I want to convey over here is that in the nucleus, in the nuclear DNA, we know that introns are there, which is around 93%. कहीं कहीं पे आपको 95% भी मिलेगा. So that's okay. So we know that this our nuclear DNA is having so much of exons, so much of introns, which is like non-coding part, right? We don't want it. Okay, but in the case of mitochondria, it does not have any introns. Now, can you justify your answer, like why the mitochondria or the chloroplast, they don't have introns? Yes, anyone who can give me the answer? Okay, something I can give you the hint, something related to the evolution. Anyone? Okay, so have you guys heard of endosymbiotic theory? Yes. Yes. So endosymbiotic theory क्या बोलता है?
That mitochondria used to be its own organism, but then it it was transported intracellularly, and it started helping the cell, and they've produced a symbiotic system there, which still survives.
हाँ. So more precisely, it was not transported, it was engulfed by another bacteria. Okay.
Yeah.
So एक cell ने दूसरे cell को खाया. So finally what happened? This is how they say that evolutionary, you know, the eukaryotic cell came into the existence where the engulfed जो अंदर चला गया था, they started acting as a, you know, energy giving source to the one which has engulfed the smaller one. Okay. So yes, so that's how the mitochondria and the chloroplast have come into the existence in the case of the eukaryotic cells. And we know that the prokaryotes, even prokaryotes, they don't have any introns. So similarly, the mitochondria and as well as the chloroplasts are not having any introns. Okay. Now look at the figure over here. Here you can see that like green shades are the protein encoding regions of the mitochondria, and rest all like yellow color things are the tRNA encoding region. So you have got specific tRNA for the mitochondrial purpose, mitochondrial protein synthesis. Okay. Now, is this much clear? There's nothing like you can see over here cytochrome B hai, NADH dehydrogenase hai, cytochrome C. Oh, sorry. Cytochrome oxidase subunits are there. So all are there except your succinate dehydrogenase. Why? Because as I mentioned, it is encoded by the nuclear gene. Rest all is over here. This is your complex 1, this is your complex 2, this is your complex, no, this is your complex, no, sorry, not complex 2, complex 3 and 4. ATP synthase subunits are also over here, right? The fifth one. So all these are part of your electron transport chain except the succinate dehydrogenase, okay, which is coming from the nuclear genes. Okay, now coming to the mitochondrial DNA replication. So there are many things which you need to remember. Let's go one by one, no hurry. Okay, so the first one. So look at this figure. Now what they have done is that they, in order to understand the mitochondrial DNA replication, this is otherwise also called D-loop replication. Okay. Many times they won't mention that this is mitochondrial or chloroplast DNA replication. It will be known as D-loop replication. Now here what they have done is that जो नीचे वाला strand है, okay, that is known as the heavy strand. This one is known as the heavy strand, okay, and they are rich in guanines. Whereas the other one, outer one, is known as the light strand. Now, heavy strand has got the origin of replication in this region. Can you see the control region or the D loop region? Okay. So these are the sequences which is meant for the heavy strand replication. और ठीक 11,000 base pair नीचे, like 11,000 base pair downstream of OH, you have got the origin for your light strand. Can you see in the tRNA encoding region? Is this much clear? Can you remember? Yes, there are so much things to remember, right? So what we need to remember is the inner strand is known as the heavy strand because it is rich in guanines. The outer strand is known as the light strand. Okay. The original replication is present in the D loop region, and origin of light strand is present in the tRNA encoding region. Okay, just itna yaad rakho. Now let's move on to the next. Okay, now in order to start this, you need a helicase. Okay, in the case of mitochondria, it is known as twinkle DNA helicase. I mean, it does sound funny, but this is the name of the helicase of the mitochondrial DNA replication. It is the twinkle DNA helicase. Okay. So after that, what will happen? A replication bubble will be formed, right? So this is how it is going to look like. Now, slowly, hey guys, are you with me? Are you able to follow?
Yes.
Okay.
Yes, ma'am.
Yeah. So this is how it will look once the DNA helicase, the twinkle DNA helicase is separating the strand. So this is how the bubble will look. Okay, replication bubble will look like. Now what is happening? The mitochondrial single-stranded binding proteins will come. Okay, and they will bind. Then what will happen? We need a RNA polymerase So it is known as polRMT. So it's a polymerase RNA of mitochondria. So it's a single subunit RNA polymerase. It is going to give you the RNA primer over here. Now, what will happen? You need a DNA polymerase. So which is the DNA polymerase here? It is the polymerase gamma. It has got the 3 subunits. And what are the activities of these subunits? It has got the polymerase activity. It has the 3-5' exonuclease activity and the lyase activity. Now, one more thing which you need to remember over here is that once the mitochondrial DNA replication is done, okay, there is no going back. What does it mean? Once the DNA replication is completed, if any error had occurred, it cannot be corrected. जैसे कि न्यूक्लियर जीन्स में क्या रहता है, न्यूक्लियर डीएनए के लिए क्या होता है, यू हैव गॉट सो मच ऑफ डीएनए रिपेयर मैकेनिज्म व्हिच विल बी डिस्कसिंग शॉर्टली, लाइक मिसमैच रिपेयर हो गया, बेस एक्सेशन हो गया, न्यूक्लियोटाइड एक्सेशन रिपेयर हो गया, लाइक मेनी मैकेनिज्म्स आर देयर इन ऑर्डर टू रिपेयर द न्यूक्लियर डीएनए. But in the case of mitochondria and chloroplasts, once the replication is completed, you cannot go back. जब आपका replication हो रहा है, then and there itself, if any mismatches are there, then you can have the 3-5' exonuclease activity. But once the replication is being done, you can't go back and go for the proofreading activity. Is this much clear? Okay. इसी वजह से, like if you have any, mitochondrial mutation, it cannot be repaired. It will be what to say, it will be passed on to the next generation. Okay, and then you must have heard about this is like a maternal inheritance, you must have studied about, right? Because the mitochondria, the offspring gets from its mother. So if the mother was carrying any mitochondrial mutation, that will be passed on to the offspring. So that's why it is known as the maternal inheritance. Whether it's a girl or a boy, it is always going to be from the mother to the offspring, correct? And that's the, like in the genetics also, we'll study more. Okay, so coming back over here. So as I said, it has got the single stranded binding proteins, goes and binds over there. Then you have got the primer over here, which is synthesized by the pol RMT. MT is for the mitochondria. R is RNA polymerase. So it's a RNA polymerase of mitochondria. Then you have got the polymerase gamma, DNA polymerase, which has got the 3 subunits. Okay. This has got the polymerase activity, 3-5' exonuclease activity, as well as the lyase activity. Okay. Now what do we see is that जैसे the polymerase starts replicating, what is happening? There is this 3 strands of DNA, correct? There is a 3 strands of DNA. Now tell me where you have seen this 3 strands of DNA anywhere else, like before we have talked about it. Do you remember? हमने कहाँ पे और देखा है ये डी लूप, हमने और कहाँ पढ़ा है?
Telomerase.
Yes, very good. So when the telomerase or not the telomerase, yeah, but the telomeres. So you must have remembered to protect the end shelter in aata hai, and then if it is required, like in the stem cells or the embryonic stem cells are there, over there telomerase is recruited. So there also when the strand is formed, okay, when the replication is happening, you can always see that the 3 strands of DNA is present, okay, and that results in the D loop formation. So whenever you see that there are 3 strands of DNA, okay, they say that it is D. D जैसा नहीं दिखता है, but it is a displacement loop, right? Because one of the strand is being displaced by the new by the new replicating one. So it's a displacement loop. It's not like as a D structure aega. It is a displacement loop. Okay, is this much clear? So what do we see here? The polymerase is replicating and it is pushing this strand. Which one is this one? The black one मैंने क्या mention किया था? This is your light one. Okay. So right now what is happening? Your heavy strand is getting replicated. So first one is your heavy strand which is getting replicated. Okay. Later the light strand will get replicated. Now please remember in the case of mitochondria, or the D loop replication, both origins will not act upon. Okay, they are not going to start the replication together. So सबसे पहले आपका heavy होगा, उसके बाद ही आपका light chain, light strand का replication होगा. Okay. Yeah, so this is like in the telomere and telomerase, we have seen the this loop formation. Okay. So as your replication is getting completed, your heavy strand is getting double stranded. Okay. And आपका जो light strand है, it is getting single stranded, and since it is single stranded, it shouldn't bind back, right? So it is covered with the help of the SSB, single stranded binding protein. Now see, अभी यहाँ पे एक और चीज क्या ध्यान देना है, अगर ये रेप्लिकेशन यहाँ पे हमने चालू किया होता, then it would have got tangled up. Okay, क्योंकि बहुत सारे प्रोटीन्स को यहाँ पे बैठना है. Are you able to imagine? See, यहाँ पे आपका हेलिकेज भी आएगा, पॉलीमरेज भी आएगा. Here also you need a polymerase. So the chances of the colliding of the polymerase is high. So that's why they don't start the replication at the same time. So once it is coming at the end, जहाँ पे आपका ये sequence जो है, it is getting replicated over here. Once it is completed, this replication is completed, then only again your primer will come, sorry, primase will come and will synthesize the primer over here. ठीक है? Clear?
Ma'am, can you repeat one more? You cut out last.
Okay. Okay. So what I was saying was that first your heavy strand is getting replicated. Okay. So as this is getting replicated, what is happening then? The light strand. Okay, this light strand is becoming single stranded. So that's why it is bound with the SSBs. Okay, single stranded binding proteins. Now, since here your twinkle DNA helicase has formed the bubble, but it did not start the replication. Okay, now it didn't start because see, if it starts, what will happen? There will be a point where the DNA polymerase on this and the DNA polymerase over here will come together. एक point ऐसा आएगा जहाँ पे ये दोनों आजू बाजू होंगे. So the chances that one of the DNA polymerase might fall off from the strand and terminate the replication is high, right? In order to avoid that, okay, what the mitochondrial DNA replication, what mitochondria do is that first it completes the heavy strand DNA replication and then only later the light strand का replication शुरू होता है. Okay, so the requirement of the proteins is like in continuous. अगर एक साथ दोनों replication शुरू हो जाएगा, so what will happen? I need proteins, I need enzymes, I need nucleotides so much in quantity at the same time. But if I do one by one, so I have got sufficient nucleotides and sufficient proteins one by one to come and complete the replication. Are you getting the point now, Shruti?
Yes, ma'am.
Yeah. Okay. So once it completes this sequence over here, okay, this is completing like this, it will go like this, right? So what is happening? यहाँ का जो sequence है वो भी complete हो गया. This is done. Now once it crosses the origin of light strand, the sequence when it crosses, okay, heavy strand ka, then your RNA polymerase is coming and synthesizing the primer, and then the polymerase gamma will come and start the replication. ठीक है, एक बार जब ये sequence cross कर लिया है, then only the light strand का DNA replication will start. Okay. Okay.
Yes, ma'am.
So this loop is formed so that it can just gives a space so that it can go and continue the replication. Okay. Yeah, and then the primer will come and then it will start the replication. Now, so termination associated sequences. So in both the cases, like your heavy strand, it is going to have the termination sequence. So where the DNA polymerase will stop the replication, and you need to have the RNA primer removal also. So here the name of the RNA primer removal is done by the RNase H1. Okay, don't get confused. वहाँ पे क्या था, यहाँ पे क्या है. Okay. Okay. Now at the end, this is how your mitochondrial DNA will look like. This is also known as hemicatenae. Okay, वो एक के अंदर एक के अंदर एक के अंदर है. Now what will happen? The topoisomerase 3 alpha, which is functioning on this, will remove this hemicatenae, and finally you are going to have the two strands of mitochondria. Sorry, two DNA of mitochondria. Okay, is this much clear? This is how your D loop replication is happening. Yes, थोड़ा सा clear हुआ? Any part, any step which was not clear to you guys? So at the end, you are going to have the mitochondrial DNA replication. Clear? Any doubts? Yeah, so I would like to stop here now. कोई doubt है तो आप पूछ सकते हो.